Thank you. Hi, hello, my name is Carla. Today I'll be presenting investigating the role of protein kinase R in antiviral activity of cyclosporine A against HCV. So cyclosporine A is an immunosuppressive drug that originally was used in clinical uses for organ transplantation, until it was discovered that this drug over there on the right, it has an antiviral effect against unrelated viruses such as those. Interestingly, its use led to the discovery of cellular proteins known as cyclophilins. Cyclophilins are a family of chaperones, and among those, cyclophilin A was the first one to be discovered. Interestingly, cyclophilin A is known to interact with PKR, or protein kinase R. Protein kinase R is an innate immune sensor that, upon sensing double-stranded RNA, it is phosphorylated, but in this case, activated. Phosphorylation of PKR leads to two different pathways. One of them is the phosphorylation of EIF2 alpha, although in this case, inactivation. EIF2 alpha is a translation factor required for protein synthesis. And when it's not working, then the protein translation complex halts, and we have aggregation of these complexes. The aggregation of these complexes lead to the formation of stress granules, which are shown as little dots in the cell that we will be seeing soon. On the other hand, PKR activation leads to the translocation of transcription factors, IRF1 and NF-kappa B, into the nucleus. Therefore, it's expected that many viruses would target PKR to prevent innate immune response. So, the first experiment that started this project is as it follows. What we did was we used HOH 7.5, cells that are usually used for HCV infection. And what we did here in the Western that you can see below is we developed two different types of cells. One of them that were expressing PKR, and another group of cells were not expressing PKR by using CRISPR technology. So we used those cells, we infected them with HCV, and after 4 hours, we treated with the cyclosporine A treatment. Now, in the graph below, as you can see in the black dots, when we have cyclosporine A treatment of infected cells, we see that cyclosporine A has a potent antiviral effect. However, in blue, in the squares, when we treat infected cells with cyclosporine A, we see that the antiviral effect of cyclosporine A is partially lost. So that meant that cyclosporine A antiviral effect is partially dependent on PKR presence. Taking that into consideration, we decided to check what would be the level of PKR activation in regular infection. So what we did was we took our cells, we infected them with HCV, and as you can see in the first lane, in the mock lane, this means that the cells were not infected, we do not have activated levels of PKR, compared to the other 3 lanes in which we do have activation of PKR due to the presence of infection. So, among all the proteins that are present in HCV virus, in the HCV genome, we decided to focus our attention in NS5A, or non-structural protein 5A. Why? Because encoded in domain 2, NS5A can interact with 1, with PKR, and 2, with cyclophilin A. Taking this into consideration, we developed our hypothesis. Our hypothesis is that cyclosporine A disrupts PKR regulation by NS5A. And the main players of this talk are NS5A, the non-structural protein, cyclosporine A, and PKR. So after that, we decided to do this experiment as it follows. We used 293 T cells, which are highly transfectable cells, and we transfected them with poly-IC, which is a double-stranded RNA mimic molecule. And I told you that PKR is activated by double-stranded RNA. So here, as you can see in the 2 first lanes, when we add poly-IC, we see PKR activation. So our model worked, and then we decided to do this. We transfected the cells with an empty vector or with BP35. BP35 was encoded in PCDNA vector, and this is a protein that inhibits very, very efficiently PKR. So after that, we decided to test if NS5A would inhibit PKR as effectively as BP35. Originally, NS5A was reported as a PKR inhibitor. However, we saw here that NS5A was not inhibiting PKR, not as efficiently as least as BP35. So these experiments were in the absence of cyclosporine A. So what we decided to do next was we transfected our cells with the empty vector. We treated with cyclosporine A, And we saw that cyclosporine A was able to reduce PKR activation. You can see it at the very top line. So after that, we decided to check what would happen if we had NS5A and cyclosporine A treatment. And we saw that in cells transfected or cells expressing NS5A, presence of cyclosporine A or treatment with cyclosporine A would not reduce PKR activation. So after that, the next experiment that we decided to do in collaboration with the Lohmann lab in Heidelberg is as it follows. In the previous experiment, we used NS5A by itself, but NS5A does not work by itself, right? It works in a complex with other proteins during infection. So thanks to Kinase funding, I was able to perform this experiment in the group. So what we had this time is also a transfection, but this time we have a complex construct that expresses from NS3 to NS5B. The catch here is that NS5A can come from either genotype 1, 2, 3, 4, or 5 from HCV. So we transfected the cells, and then we checked what would be the level of PKR activation or stress transformation. And what we saw is not much change. NS5A, regardless of the genotype it came from, and this time working as a complex, still was not able to inhibit PKR activation. So considering this, we thought that perhaps transfection itself, like adding a vector encoding NS5A, could cause PKR activation itself, as you can see in the GFP channel. So what we decided to do was develop a bit of a more pure model. So in this pure model, we have cells that we could induce NS5A expression from genotype 1B by adding doxycycline treatment. This is a complex method, but pretty much the takeout is that if I do not add doxycycline, my cells do not express NS5A. But if I add doxycycline, my cells express NS5A. And you can see it in that Western over there. So doxy 4 micrograms, minus no NS5A flag, plus NS5A flag. So here I'm demonstrating that I can express NS5A here. However, when we treat with transfect with poly-IC, we see that PKR activation is not inhibited by NS5A presence. So in this working model, we saw that PKR activation here, PKR phosphorylation, was not really being affected. But we decided to check what would happen with stress granules formation, which is down below here, below G3BP1. And what we did was this immunofluorescence. So let's focus first on the green channel. The green channel, I hope you can see, is NS5A. So cells that are green are cells that are expressing NS5A. So on the panel above, no doxy, no green, right? Because I'm not inducing NS5A expression. However, in the panel below, we do have NS5A expression because we added doxycycline. So doxy-inducible NS5A, I see green. Now, on the second, in red, that's G3BP1. I stained for G3BP1. G3BP1 is a marker of stress granules formation. And we see stress granules in little red dots, or punctua, aggregated in the cell, which you can see in the first red panel above. However, we saw that poly-IC would induce stress granules formation in cells that were not expressing NS5A. However, when we had NS5A expression, we saw that cells expressing NS5A would not have stress granules formation. And we saw, okay, that's kind of interesting. What will happen if we add cyclosporine A treatment? So what we did was the following. We induced NS5A, treated with poly-IC as always, and this time we treated with cyclosporine A. Now here in green, you can see that the cells that are expressing NS5A also express stress granules. Only the ones at the front will be able to see it. The ones at the back, I'm very sorry, but not too much. But yeah, point is that we saw that cyclosporine A could rescue stress granules formation in NS5A expressing cells. So the conclusions and the significance of this project were that antiviral effect of cyclosporine A relies partially on PKR. NS5A expression does not inhibit PKR activation. NS5A may reduce stress granules formation, and cyclosporine A rescues stress granules formation in NS5A expressing cells. Future works in PKR activation or PKR regulation are important because hepatocellular carcinoma patients present activated levels of PKR in surrounding tissue. And also, in vivo studies with mice, we have seen, well, others have seen, that PKR inhibition in the liver improves liver disease survival. So also further studies from our part would be focusing on what would be the state or what would happen with IRF1 and NF-kappa B in the presence of cyclosporine A. And, oh, I made it in time. Okay, great. That's all. Thank you very much. That's it.
Thank you, Carla. I think there's time for one quick question, if anybody has a burning question. Jordan?
Yeah. Thanks, Carla. Very nice presentation. Just a quick question. I guess the previous data showing that NS5A inhibited PKR, what models were they using and how did it differ from what you were doing?
Oh, okay. So this was very long time ago, and it was not performed in a cell culture. It was performed in an enzymatic assay. So it was more chemically it was not performed in the cell, so it's like you're not considering variables that you would usually see in an actual infection or in an actual living model,
And
in this case, cell line.
And would your effect on the stress granule production, I guess, would that have been evident in the enzymatic assay?
No, because the enzymatic assay was just, let's say, the two proteins, yeah. Okay, whereas here in the cell, you need a marker, and in this case, it's more dynamic. So for us, it's G3BP1, but it can also be TL1. Yeah, you can only see it in the cell. I don't think you can see it in the model, because in the model, you don't have the translation complexes.
So you think the old data was just wrong because it was a simple assay? That's fine. You can say that. You can say that. Yeah, it's important because that's sort of a long-standing thought that NS5A inhibits PKR activation. So your data show that it doesn't, which is important. So
yeah.
I think we've spent like 3 years convincing ourselves that our data is
yeah, we were like, we're going against the filters. Yeah.
Okay. So thank you, and I won't keep you from the coffee break. So we'll go to coffee break and come back here at 10 o'clock for the next session.